So for part three, now we're going to have a look at another two functional groups that both have quite a lot of biological significance. So firstly, we'll be having a look at the ester functional group and its role in connecting an important lipid called triacylglycerol, which is actually held together by ester connections. And we'll also be looking at the phenol functional group, which has an interesting role in mitigating against free radicals when you have a phenol compound like vitamin E or polyphenol that can act as an antioxidant. So let's start off with the ester functional group. So the ester functional group looks a little similar to a carboxylic acid functional group without the hydrogen. And by losing the hydrogen, it can actually attach to an extra R group. So this is another one of our two R group functional groups, and it's also got a carbonyl, this time attached to an oxygen. And because the oxygen needs to form a second covalent bond, there can be a second R group. So we can either draw it out like this with all the bonds, or you can have it written as COO, and then we've got the R on either side. So we can say the ester functional group is a carbonyl attached to an oxygen and two R groups. So the ester compounds will have a noate at the end of their name. So for example, compound like methylpropanoate will be an ester containing functional group. So here we can see the structure of methylpropanoate. So let's circle the functional group atoms, which are in the center here. So because it's a 2R group functional group, we always have the functional group atoms in the middle of a compound. Then we've got one R group on the side here. So the CH3 is one of the R groups. That's the methyl part. And on the other side, we've got two carbons, which in combination with the carbon in the carbonyl corresponds to this prop part. So that's three carbons there. But really all I want you to be able to do with this sort of structure is recognize where the functional group is and what it is, and the fact that methylpropanoate has the ester functional group within it. So thinking back to the difference between a 1R group or 2R group functional group, so we saw quite a few 1R group functional groups today, like the aldehyde or carboxylic acid. So these are always at the end of a compound or at a branch point. Whereas a 2R group functional group, like the ester or the ketone functional group, actually is going to be present in the middle of a compound. Because remember, the R tells you how many connections to other atoms there will be that are not a part of the functional group. So the ROH designates that there's going to be one connection, whereas for a functional group like the ether, where there's two R groups, you've got the functional group atom that's bonded to two different R groups, so two different sets of atoms. So we saw on the previous slide that we have the ester functional group in the center and then the R groups on either side. So we can class the ester functional group as a 2R group functional group. So here we can see an example of a 1R group compound where we've got the alcohol up top and the rest of the compound is considered the R group. Whereas for the ether, we've got the oxygen atom in the middle and R groups on either side. So let's have a look at how this 2R group functional group is actually allowing the connections within a triacylglycerol compound. So the fats and oils in our diet are actually mostly made up of this lipid called triacylglycerol. And a triacylglycerol is actually a combination of 3 fatty acid compounds and 1 glycerol compound. So let's have a look at those individual structures. So glycerol at the top here contains 3 alcohol functional groups. So you've got an OH in each of these positions that I've underlined, and the rest of it is a carbon chain. So because the glycerol has these 3 alcohol functional groups, essentially that means that there's 3 positions that it can form a chemical reaction with another compound at. So each of these can be involved in a different chemical reaction. And what about the other piece that we have present in triacylglycerol? A fatty acid. So as its name suggests, it's an acid. And do we recognize the functional group here? We've got the carbonyl and an OH, which corresponds to the carboxylic acid functional group, our only acidic functional group. And that carbon within the fatty acid functional group is actually connected to many other carbons and hydrogens, as shown by the line bonding formula of this zigzag pattern. So remember, each position within this line bonding formula, there's carbons and their associated hydrogens attached. So whenever we see this zigzag pattern, you know there's lots of carbons in a row, and it's common representation we see for our fatty acids that contain these long chains of carbon and hydrogen atoms. So for a triacyl glycerol compound, we've actually got 3 of these fatty acids and 1 glycerol connected together. And we can see that in the structure on the right hand side here. There's the fatty acids, so there's 1 here, and 1 here. And on the left hand side is our glycerol. Okay, so how are these 2 compounds connected? Well, you can see that in between the compounds, or connecting the compounds, is this group of atoms that I'm putting the box around. So does the group of atoms that I'm putting the box around here correspond to any of the functional groups we've seen? Well, it's actually the ester functional group. So within this group of atoms, we've got our carbonyl, C double bond O, and we've got another oxygen and the R group. So in this case, the R groups are the rest of the fatty acid on one side, and on the other side is the rest of the glycerol compound. So it just shows you that a 2R group functional group can actually be used to connect different compounds together. So in this case, we're connecting 3 fatty acids to 1 glycerol by forming how many of these ester functional groups? 3, because each ester functional group is responsible for attaching 1 fatty acid to the 1 glycerol. Remember, the glycerol has 3 alcohol groups, so it can take on or attach to 3 different fatty acids. So we've got this ratio of 3 to 1. So where might you find a triacyl glycerol, or what is a triacyl glycerol? Well, basically it's the main component of our fats and oils in our diet, so it's a very common compound that we see in the body. So the triacyl glycerol can either be broken down in the body to be converted into cellular energy through metabolic processes, and it's also possible for us to build these compounds in the body and store them as fat in the adipose tissue. So here we can see the alcohol functional group within glycerol in the red circle, carboxylic acid functional group in the orange, and then these 2 fuse together to make our ester functional group as shown here in blue. So we've got an ester functional group connecting each of the fatty acids to one of the alcohol groups within the glycerol compound. So as well as being able to make the ester functional group, it's actually also possible to break it down. So in this representation here, we can see that we can interchange between having the compounds separate, so glycerol and 3 fatty acids separate, or they can be fused together in a triacyl glycerol. And when we do this fusion of the functional groups, it actually releases water as well, which I'll show you in the following slides. So what I was showing you in the previous slide is the process of creating esters in terms of using glycerol and fatty acids to connect together to make the TAG, triacyl glycerol. And we would consider this process esterification, or put more simply, creation of an ester. But if we're going to remove those ester functional groups from the triacyl glycerol compound, we call this ester hydrolysis, or breaking down of the ester. So what would the consequence of breaking down the ester groups within triacyl glycerol be? What would this do to the compound? It would split each individual compound into its individual pieces. So instead of all being together in one unit, they'd be split into individual compounds. So that's where you would get a single glycerol and 3 fatty acids instead of them all being together in the TAG. So in the esterification process, essentially we're connecting fatty acids to glycerol, and this is what's required to generate triacylglycerol. So this does happen in the body, and when more triacylglycerols are being created in cells, they can actually be stored as fat. So there are specialized fat storage cells called adipocytes, which are present in the adipose tissue. So ester hydrolysis is the reverse of this process, where we can actually remove ester groups or break them down. And this will mean that the TAG will split into the individual compounds. So instead of being one complete unit, we now have 3 individual fatty acids and 1 glycerol. And this process has to occur before the triacylglycerol can be converted into cellular energy. So there are reactions that allow glycerol and also the fatty acids to actually be converted into ATP. So when the triacylglycerol is needing to be used as an energy source, we can actually undergo specific metabolic reactions to make this possible. But the first step is to remove the ester functional groups to basically split the triacylglycerol apart. So here's an example of a esterification reaction. So this is creating an ester. So in the summary here, we can see that when we have an alcohol and a carboxylic acid, they can combine together to create an ester functional group. And as they do this, some of the atoms within the functional group need to actually be released, because that's the only way we can create the new bonds that are present in the ester. So when the alcohol and carboxylic acid release some of their atoms, there's actually enough atoms to make a water molecule. And that's why the water molecule is the product of the reaction. So starting off with an alcohol compound, which is methanol. Here we can see the alcohol group here. And another compound, this time it's going to be a carboxylic acid compound, ethanoic acid. So we've got that carboxylic acid there. So as it shows you above, we're going to get these two compounds, functional groups, reacting together to create a new functional group. And we can see that on the right hand side here. So we've got a combination of these two functional groups on the left coming together to make our new ester functional group. So how is it possible for these functional groups to come together to make a new functional group? Well, it goes back to an old saying I had in an earlier lecture. You got to break bonds to make bonds. So at the start of the chemical reaction, we need to break some of these chemical bonds to allow there to be space to form new bonds. So the bonds that are going to be broken are actually the bond between oxygen and hydrogen within the alcohol group, and the bond between the carbonyl and the OH group within the carboxylic acid. Those atoms that are lost actually come together to make water. So water is a combination of an OH group and also a hydrogen atom, and they come from these atoms that have been lost from the initial compounds. And once those atoms have been lost, the two individual pieces that are left over from the methanol and the ethanoic acids can actually come together and attach to form a new covalent bond. And when it does this, we've got this new functional group, which is the ester. And remember, because it's an ester, it's got the two R groups on either side. So it's connecting the two individual compounds that we see on the left-hand side to now be together in one single compound. Alright, now let's have a look at an animation of how the ester is actually formed in this esterification reaction. So we've got our methanol and our ethanoic acid with the functional groups that are going to react to create the ester. So initially we're going to get bond breakage between the hydrogen and the oxygen in methanol, and between the OH and the carbonyl in ethanoic acid. So once those bonds are broken, we then have those individual atoms that were part of the compounds coming together, and what will two hydrogens and an oxygen form? They're going to form a water molecule. But that leaves two remnants of the compounds here without the maximum number of bonds, or the necessary number of bonds to actually be stable. So which atoms here need to have more bonds? Well, this oxygen within the compound on the left needs another bond, and so does this carbon within the carbonyl group here. So what's the solution? Well, if these two compounds move close enough together, we can get this new bond forming to satisfy the bonding arrangements of both atoms. And we can see that new bond shown in purple here. So by creating this new bond, what functional group has been created in the methyl ethanoate? We've now got an ester functional group. So essentially what's happening in this reaction is the methanol is losing a hydrogen and the ethanoic acid is losing an OH group, which come together to make water. And then to satisfy the bonding arrangements of the atoms that are left over, we then get the carbonyl carbon forming a covalent bond to the oxygen within the other compound to make a new compound, which is the methyl ethanoate containing the ester. So essentially by doing this reaction, we're converting the two carbon containing compounds into a single carbon containing compound that is larger. So it's being fused together by that new ester functional group there in blue. So while esters can be made by esterification, it's also possible to break them down through a reaction of ester hydrolysis. So when might it be useful to break down an ester? Well, if you want to process the compound that contains the ester, the first step is to break the ester, and then the individual compounds can be used for other things. For example, fatty acid can be used to make cellular energy. So in this way, we're going from an ester compound and converting it into one alcohol and one carboxylic acid compound. But to be able to regenerate those alcohol and carboxylic acid compounds, this time we actually have to add in a water molecule. So a water molecule is actually the other reactant that's required in the reaction. So the first thing that's going to happen in this reaction is to actually break the bond within the ester functional group. And this will allow the two pieces that were coming together in the ester functional group to actually separate. And once they separate, we can have the functional groups being remade by forming an additional chemical bond. So by the oxygen forming a bond to hydrogen, we regenerate the hydrogen atom, and by the carbonyl group forming an additional covalent bond to the OH group, we've now got the carboxylic acid. So these new atoms that are being added in, the OH here and the hydrogen here, where did they come from? Well, they came from the water molecule. So essentially the water molecule will help break down the ester compound by allowing the oxygen and the carbon in these two structures to actually be stable by having the correct number of bonds. So let's see how this process works here with our animation. So the first thing that will happen is the water molecule will come in and basically attack that covalent bond that is attaching the oxygen and carbon together within the ester functional group. So we get the breakage of that functional group, breakage of the bond between the oxygen and carbon, and that allows the two compounds to separate. And after that occurs, the water molecule will actually split in two as well, and that allows the OH group to form a new bond to the carbonyl to satisfy the bonding arrangements there. And we're also going to get a new bond between the other hydrogen that was a part of water and the oxygen there. So you can see by doing this, essentially what was one large compound has been split in two. And we can only do that because the water molecule is satisfying the bonding arrangements of the other atoms involved that had been connected within the ester functional group. So essentially by breaking the ester functional group, we then are able to recreate the functional groups of the alcohol within the methanol compound and the carboxylic acid functional group within the ethanoic acid. So another participation plus for you guys to consider to try and work out how the ester functional group is related to our triacylglycerol and why it's useful, and how the esters can be formed and also broken down. So make sure you have a go at this question to check your understanding of this section of the lecture, and also be sure to share your answers with your peers to create a discussion and collect ideas about what you're learning. So we've also reached the point where you can have a go at another 2 of the revision questions in the revision quiz. So 7 and 8, have a go at those now. So please pause and have a go at those questions.